Good morning, good afternoon, good evening, everyone. I understand there are folks on the line from all around the world, so welcome to Electrophoresis. My name is Mitch Holland, and I'm a professor of biochemistry and molecular biology at Penn State University. I'm also a forensic scientist, so I came to Penn State to teach forensic science. Let's see, okay. So I'm going to talk to you today about electrophoresis. And electrophoresis sounds like something technical and really kind of a boring topic, but it's something that if you do any science in the lab related to DNA or proteins, you're going to do some form of electrophoresis. And it's pretty cool. So you can see, electrophoresis. Electro is just electrical energy. So you apply an electrical charge across a surface, and then that protein or DNA moves through that continuum of space and will separate. And depending upon whether you're separating on shape or size of the protein or the piece of DNA, you can get some information about it. And so when we think about electrophoresis, we're thinking about obstacles in space, right? So when you have any electrophoretic environment, there's some space where the DNA or the protein can move through, and then there's some obstacles. So this is a classroom, right? Everybody's familiar with a classroom. As you look at it, these desks are pretty close together. So trying to navigate through those obstacles is pretty tough. You can go down the rows, but you really can't go across the rows. And so you'd have to come back and then come back down again. So this is a lot of obstacles and very little space, hard for proteins and DNA to move through, versus this space here, right? This space has a lot of room to move around in different directions. And so this is probably more reflective of what an electrophoretic system looks like, navigating through those spaces and past those obstacles. And then one of the questions is, what's pulling you through the space, right? How do you get from one end to the other? If you've walked into the classroom and you're in this corner, what's pulling you to the other side? It may be sitting down at a desk. It might be a big chocolate cake on the other end and everybody's trying to get to the chocolate cake. If it's just one of you, right, you can move pretty quickly through that space and navigate around those desks and get to the cake. But if 4 or 5 of you lock arms and try to navigate through all that space, right, it's going to be harder for all 5 of you to get through that space. So bigger pieces of DNA, bigger proteins will go slower because they have to find more space to navigate through and they bump into things more often. So that's the basic principle of the separation process of electrophoresis. Now, I kind of think of it more as a Plinko environment where you start with your DNA and protein on one end and it starts to move through your obstacles. And maybe you've seen the show The Wall, which is a game show we have in the United States where they play this game and answer questions. And these balls go down through the Plinko machine and go into slots that give them lots and lots of money or take away lots and lots of money. But the way we get the balls through the Plinko is just by gravity, right? You release them and it just falls because of gravity. Well, typically when we're doing electrophoresis, we can't use gravity. We have to apply some sort of force to move those molecules through the obstacles. And again, typically, if you're in a molecular biology lab, you're going to be looking at DNA. So this is the structure of DNA. You may have seen it before. What's most important is that you have these base pairs that link things together and allow you to sequence the DNA and understand the genetic code. But then you also have this backbone here that's a bunch of phosphates, and phosphates are negatively charged. And so actually for every unit of DNA, right, the genetic code of A, G, T, and C, each of those letters or units has a charge associated with it. So there's a constant charge to mass ratio there that allows us to use an electrical charge to move the DNA through a system like this. Now, if we didn't have any obstacles, everything would move at the same pace because you're bigger, but you have more charge, and so you move just as fast as a small molecule with less charge. So we need obstacles, right? We need a sieving matrix. We need something to block us. We need those obstacles. And those obstacles, again, allow the DNA molecule to come through and navigate past those obstacles. And if you're a small piece of DNA, it's easier to navigate. You can kind of clump up into a ball and move through pretty quickly. But if you're a big piece of DNA, it takes a little time to get through. And sometimes, right, different portions of the DNA molecule are bunched up, and sometimes they're elongated. So it takes a while for a DNA molecule to figure out how to navigate through those obstacles. And the sort of classic electrophoretic system that you'll see maybe in high school, certainly in college if you're a scientist and you're interested in this area, agarose gels. Agarose is a natural polymer. It's a polysaccharide. And all you do is you add a little dry agarose to some water, heat it up, cool it back down, and it solidifies into this gel. And then this is a series of slides that came from the website that your host here is going to provide you. And it's hosted by the Amoeba sisters. And so the Amoeba sisters have this series of YouTube videos that talk about science, and they're pretty good. So this is their view of the agarose gel. And you can see that there are ends here. This is an end, and there's another end, and then there are these depressions here. And those are wells. And so you load your sample into those wells. In this case, it's indicating a DNA sample. You then apply the electrical charge across the gel, and the charge goes from a negative end to a positive end. And that makes sense because DNA is highly negatively charged and it wants to move towards the positive end. It's attracted to the positive end, right? Negative and positives are attracted to each other. And when they pass through, they separate out by size because of those obstacles. The bigger pieces take longer to go through, and the smaller pieces take less time to go through, or go through easier. So you get these banding patterns of pieces of DNA. And as a forensic scientist, right, this is what we do. We look at DNA from a crime scene here. We have a banding pattern, and then we have 3 people who could have left that DNA at the crime scene. So who wants to tell me which of the suspects is the one who's in trouble?
Thumbs up if you guys think you know. Or Miss Delgado's class, you can have your hands up. We can see you guys there.
So I see.
Oh, we got one right here. Here, I'll unmute her. So Lorianne, do you want to unmute and tell us?
I think it's suspect number 2.
Suspect number 2, excellent. Right, the banding pattern between the crime scene evidence and suspect 2 matches all the way across the length of the gel here. And that's a very simple view of what we do in forensic science. We don't use gels anymore. Gels are pretty old from the perspective of the technology used nowadays in forensic labs. We now use what's called capillary electrophoresis. And capillaries are really thin fibers almost. They're almost like optical fibers. This is a capillary array. So each one of these really thin glass fibers, the DNA will move through those fibers and come past the detection window. And when they come back past the detection window, they'll be picked up by a laser. The laser will excite some dyes. And these dyes are kind of like flashlights. So you excite them or you turn them on, and they turn on as a red flashlight or a green flashlight or a yellow flashlight or a blue flashlight. And they tell you what pieces of DNA are moving through that detection window. And this is what a forensic DNA profile then looks like. So this is, as we look at it, if we just look at the macro view, right, there's some blue peaks here or spikes, right? They look like spikes. These are just the bands of DNA that you saw in the gel, but instead of the view of them as bands, the relative intensity of those bands is converted into a peak. So these are just bands of DNA across a blue channel where we have blue light. Here are green pieces of DNA. These are yellow. They're marked in black because you can't really see the yellow. And then red in the 4th one down, and then purple. So this is a forensic DNA profile. If you have this profile and it matches the crime scene, that's your DNA, to the exclusion of everyone else in the world, with the exception of identical twins, right? If you have an identical twin, you'll have the same DNA profile. Otherwise, you will not have the same DNA profile of anybody else in the world. It's that powerful. Now, if we zoom in a little bit, you can kind of see what these pieces of DNA look like. So the first two peaks here are indicating X and Y. That's telling me that the DNA associated with the sample is from a male individual. Males have an X chromosome and a Y chromosome. So this is from a male individual. And then I move down to the next position here, and there are two more peaks. And when we think about these peaks, we're looking at the numbers of 17 and 18. The reason these peaks are separated by size is that they have repeated regions of DNA that will repeat a certain number of times. So in this case, we have a 17, there's 17 repeats, and then there's 18 repeats. At the next location across the genome, we have 12 repeats and 13 repeats. So the DNA profile is simply 17, 18 and 12, 13. Relatively simple to decode the DNA profile. Now, and the reason we have two in many cases is because we inherit pieces of DNA from our mom and our dad, right? So we inherit perhaps the 17 from our mom and the 18 from our dad. And that profile now is us. Versus when we look at our mom and our dad, they're going to have half of our DNA, right? But they won't have the complete same DNA because they came from different moms and dads. They came from your grandparents. So we use this information again, just like you saw in the gel image, to compare to references from victims or potential perpetrators of crime, and we'll make matches and then report back to the police and say that it seems like you have your person, and they then move to the courtroom and have a trial. So, but without electrophoresis, we can't do this. And so electrophoresis is something that is a core principle, core tool that we have in science and molecular biology in particular. So I'd be happy to answer any questions about any of that. We can back up and look at some slides, whatever you guys want to talk about.
Absolutely, and I'll help with the Q&A. Just 1 second here. Those of you emailed me some questions, but those of you who didn't email me some questions, the chat is open and you can send them by way of chat. But one of the email questions, they want to know, where do you see technology headed in regards to supporting what you do, what people do in your profession?
It's a great question, and it's really headed more towards sequencing DNA now, rather than looking at length-based differences, right? All we talked about there were just separating pieces of DNA out by length. These days, everyone's moving now towards sequencing pieces of DNA. And so you can get the length information, right? You sequence it, you know it's a certain length, but you can also get individual sequence pieces of information. And the new technology is allowing us to do things that is sort of crazy. We used to have to do every test separately. So we do test 1 and test 2 and test 3. We do them all separately. Now we can do not only those 3 tests, but we can do 400 tests all at the same time. So our ability to get information out of our DNA these days is been revolutionized over the last 10 years. And for you guys, you'll go to the doctor someday and you'll get blood drawn, and they'll take it to the lab and they'll do a panel of assessments based on your biochemical makeup. And they'll tell you, well, you have to be careful not to eat too much ice cream because the fats in the ice cream are going to do this to you, because we've looked at your DNA profile and we know that you have a predisposition for taking on too much of that ice cream fat. Of course, there'll be things that are much more important than that, like diagnosing different conditions or diseases that will help you out in terms of helping you to not be unaware of that, and therefore you can plan for it and try to do things that will stop it from occurring either earlier or at all.
One of the kids emailed me and they wanted to know, and I'll kind of paraphrase it because I don't have it in front of me, but they wanted to know what other professions besides criminal justice has this electrophoresis kind of touched on, because we know that there's all different kinds of like DNA sampling for like ancestry and all that kind of stuff. Do you see it kind of headed in the direction of other industries?
Yeah, I mean, again, anything where you're working with proteins or DNA, any industry, whether it's medical, genetics, commercial companies developing pharmaceuticals, whatever it might be, anything that involves proteins and DNA, you're going to do probably some form of electrophoresis. So it's a really important thing to sort of understand and know, because you're going to use it most likely.
Absolutely. And what kind of advice do you have for all the students around the world as far as what kind of things should they be studying? I know we have some younger kids who are in middle school versus high school and versus college. Like, what areas should they kind of focus a little bit more on?
Well, you know, if you're interested in science, obviously you have to have an interest in science in order to go down this path and be exposed to electrophoresis. But if you're interested in science, the foundational elements of science come from chemistry. So you really need to know chemistry, right? You need to know the atomic structure, the molecular structure of things, so that you can appreciate how these things perhaps interact with each other. And so chemistry really is foundational. You need some idea of biological systems. So it can be microbial versus human, right? It doesn't have to be just human. But something about biological systems, and then you combine that chemistry and biology, and then you can utilize it in different ways. You can become an analytical chemist, or you can become a physical biochemist, but you really need that chemistry and that biology mix to do the things you probably want to do, medicine, whatever it might be in the sciences.
Absolutely. And before we let you go, I know we asked you this question before, but I know we have a lot of new students who are on here. But is there any advice that you could give to all of the students as they go out there and figure out what they want to do, even if it doesn't have to do with criminal forensics?
Yeah, I remember you asking me that question, and I remember the answer, which is explore, right? Poke around. Look at, attend a talk like this and say, oh, electrophoresis, that sounds really cool, or oof, that was pretty boring. I'm not really interested in electrophoresis, right? So explore, go out and read something or attend something, see what's out there. That's my best advice. Whenever I talk to students in my classes or that come in for advice, I always tell them, take a class in something that you think you might be interested in. When you take the class, you might find that you're just wildly interested and that's it, and you go down that path, or you may find, nope, it's not it, we'll try something else.